Hello students. Yesterday we had done the principles of inheritance, that is laws of inheritance that was given by Mendel, law of dominance and law of segregation. Now coming to the next topic, that is incomplete dominance. Now Mendel had done some experiments and he gave his laws, but later there were many flaws or many mistakes that were found out by other scientists. So after some experiments that was done, it was found that there is a phenomena called incomplete dominance. Now actually Mendel said that if a tall and a short plant they are crossed, either tall or short, these are the two phenotypes that are obtained. But later it was found that it's not like that. An intermediate feature is also obtained. So look at this incomplete dominance. It is, you can see here, a white flower and a red flower, and what is the offspring that is obtained? That is a pink. So this is not neither the one which was neither of the features that was present in the parents, that is what we are getting. So this is an intermediate phenotype. So this is what is called incomplete dominance, that is appearance of an intermediate phenotype in a heterozygous organism obtained when two homozygous parents of different phenotypes are crossed. That means suppose this is white WW and small w, then you are getting capital W small w, but the color is pink. Okay, so that is incomplete dominance. Take another example of incomplete dominance. You can see here here R and small r. According to Mendel, capital R small r will be giving red color, but here it is found that it is pink color. So this is called incomplete dominance. You can see the Punnett square here, R. Here this is red, purple and white. So 1:2:1. This is the phenotype. Whereas in Mendel's experiment it was found that the phenotype is 3:1. So here 1:2:1, that is one red, two pink and one white. Now this was the explanation that he gave, that was given for the concept of dominance. Every gene contains the information to express a particular trait. So that every gene will be expressing a particular trait, and in a diploid organism there are two copies of each gene, that is as a pair of alleles. These two alleles need not be identical as in a heterozygote. One of them may be different due to some changes that it has undergone, which modifies the information that particular allele contains. So that modification that is taking place, that is what is expressed as another feature. So that is what is called incomplete dominance. Another example of incomplete dominance is the gene. Consider a gene that contains information for producing an enzyme. So this enzyme, what it will do, this is needed for the transformation of a substrate S. Okay, suppose let us assume that the normal allele produces the normal enzyme that is needed for the transformation of a substrate S. Now there are two copies of this gene. Now normal allele is producing enzyme for the transformation of a substrate S. Now modified allele, what would modified allele do? It could be responsible for production of normal or less efficient enzyme, non-functional enzyme or no enzyme at all. Now in the first case, that is the normal or less efficient enzyme, the modified allele is equivalent to the unmodified allele. So in this case, this is equal to the normal enzyme. Unmodified allele, that is it will produce the same phenotype, that is it result in the formation of substrate S. This enzyme will result in the formation of S. Then there is no modification, and this is the dominant allele. Now considering the second and third case, in the second and third case the phenotype will be affected. The phenotype will only be dependent on the functioning of the unmodified allele. So this will become recessive, and unfunctional and no enzyme, that will become recessive, and unmodified allele will be the dominant. So the modified allele is the recessive allele, and the recessive trait is seen due to non-functional or because no enzyme is produced. So here you see the normal allele is the dominant, and it will help in the transformation of a substrate. It will produce an enzyme which help in the transformation of substrate, whereas this modified allele is the recessive allele. It will not help in the production of enzyme that help in the transformation of substrate. So that is the example of incomplete dominance. Now coming to codominance, another topic here, codominance. Here you had an incomplete dominance, you had seen there is an intermediate phenotype that is occurring. Now codominance, the best example of codominance is human ABO blood grouping. Now what happens here is appearance of a mixed phenotype in a heterozygous obtained when two homozygous parent of different phenotypes are crossed. This will be clear when we take the example. Now here both are dominating. In ABO both are dominating. In codominance both the parents they become dominating. For example, human ABO blood group. Now human ABO blood group, they are controlled by gene I. Now this gene I has 3 alleles. Now IA, IB and IA. Till now you had seen 2 alleles. Here you are seeing 3 alleles. This is called multiple alleles. So IA, IB and I. Now all the alleles they control the synthesis of proteins. IA produces sugar. IB produces sugar of a different type. And small I and this I does not produce any sugar. The small I that does not produce any sugar. Now this is what is written as small I. In the table which will be seen. Now each individual has any two of these 3 alleles. Now example of you can see this table here. This table shows the blood groups in human population. Now you can see here parent from 1 IA, parent 2 IA, allele from parent A and parent 2 is IA, and the genotype that you get is IA. What is the blood group there? A. Here you see IAIB. Genotype is IAIB. Blood group is AB. IA small I. Genotype IA small I. Blood group is A because A is dominant here. IB, IA, IAB, AB is coming. So this is what is called codominance. Here A and B both are being expressed. So this is the example of codominance. IB, here IB, so blood group is B. IB small I, here B is dominating, so B is the blood group. Small I, both are recessive, so O. That is the blood group O. Now this is what is called codominance because here you have some people are having blood group AB. Why? Because A and B both are dominating. This is called codominant. So IA and IB are codominant with each other. This is because both are being expressed, they are codominant. This is called codominance. Whereas IA and IB are completely dominant over small I. Their dominant IA and B are completely dominant over small I. Whereas IA and IB are codominant. So this is a best example of codominance. So how many phenotypes are obtained? AB, AB and O. Now genotypes, how many genotypes? 6 unique combinations you get. That is IAIB, IAIA, IBIB, IAIB, IAI, IBI and small I. So what I told earlier, multiple alleles, it is a non-Mendelian inheritance pattern and there are genes which have more than 2 alleles. So multiple alleles of ABO is IA, IB, small I, and there are more than 2, that is 3 alleles governing the same character. So here IA, IB and I, they all are governing the character for our blood group. So this is the example of multiple alleles. So this is what is done in codominance and incomplete dominance.